Everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Eugenia and I'll be your host. It's our pleasure to have LTS with us today, who will be presenting this webinar titled OBDS on Body Delivery Systems. Today's guest speaker is Greg Moakes, Executive Vice President, Business Development, Sorrel Platform at LTS. Greg is an analytical chemist by training. He received his PhD in chemistry from Georgia Tech and later his MBA from Southern Methodist University in Dallas. He started his career working in the specialty materials business, spending time in global procurement, innovation leadership, sales leadership, and business development roles. Greg has over 15 years experience in launching products in the biomaterials, medical polymers, and drug-eluting implant fields. Greg currently leads commercial activities for the Sorrel wearable drug delivery device platform. He is passionate about partnering with pharmaceutical companies to ensure their combination products give the most intuitive, stress-free experience for patients. Now I'll provide some housekeeping information before we start. First of all, I would personally like to welcome you to our webinar platform On24. You will notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. During today's event, there will be plenty of time to ask our speaker questions. You can do that via the questions widget on the left-hand side of your screen. Just type your question in this box and click Submit. We will allocate some time at the end of the session to answer questions or any thoughts that you may have. We're having 3 short polling questions today, and you can vote simply by selecting the answers relevant to you. We would also like to invite you to fill out a short survey, which you can find in the middle of the screen. Excuse me. If you require any assistance, please use the yellow Help widget. In addition, you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. And now, please allow me to welcome Greg. Over to you.
Thank you, Eugenia. And as you mentioned, good morning, good afternoon, good evening, depending on where you're joining us from. Today, I will be presenting the LTS Device Technologies Sorrel Platform, and specifically answering the question, how do we solve the dilemma of essentially maintaining a very simple process at the pharmaco when it comes to combination products versus the simplicity of patient use. So thank you for the introduction. Again, my name is Greg Moakes. I'm an analytical chemist by training. I spent 15 years in the biomaterials, the biopolymers, and most recently leading a business development team in solid implant dosage forms. And that was up until around a year ago when I joined the Sorrel team at the time with A10 Medical. And what a year it's been, not least of which because we were very fortunate to be acquired by LTS Lohmann this year. And if you look at some of the numbers there, we are poised to make investment to turbocharge the growth and our ability to help pharmaco partners deliver combination products effectively and safely. Not least, my excitement is not least because we now have a global footprint. So very healthy global footprint via LTS, and this allows us to think about future expansion of both manufacturing and R&D. And very excited. We're all very excited on the Sorrel team. So before we get into the solution, why a wearable for drug delivery? And I think it's important to start at the beginning and say, why an injectable? Well, the vast majority of the R&D pipeline, if you take a look now, are biologics, and they are not deliverable in large by traditional methods such as inhalation, oral, for example. So the traditional routes do not work in large part for biologics, and it drives the need for an injectable. The injectables that are currently available to us would be IV inpatient infusions and subcutaneous infusion via needle and vial, via auto injectors, and so on and so forth. But the trends in the treatment of chronic diseases are to drive people out of the healthcare environment. If COVID taught us one thing, it's that we should work towards less reliance on inpatient treatments and move more towards the comfort and the convenience of the patient's home. So you see a lot of the pipeline being reformulated from IV to sub-Q, and you see a lot of the new molecular entities being biologics and perhaps going to subcutaneous first now because people are aware of this trend. We want to get out of hospitals wherever possible. The established injection technologies were fit for purpose for a while, but they have volume limitations, and the volumes of biologics delivery are creeping up. So you've got volume limitations, and as the volumes creep up, you're asking the user to hold essentially a pen against their skin for a longer amount of time, and it's becoming unacceptable. And that drives people towards a wearable solution. There are several wearable solutions that were commercialized. I think three commercial products to date on the market. And moreover, the volumes are starting to hit 20 milliliters and above. And within that space, I think we have one competitor, but there are very few options for large volume on-body wearable systems above 20 milliliters. So there are several key kind of guiding factors that we use when we're developing an effective on-body delivery system. These are things that we believe are very important to our pharmaco and to the end user. We don't think we should be asking our pharmaco partners to choose primary container. If they have a cartridge from a previous project, fine. If they have a vial that they've been using for decades, very, very standard ISO vials in the pharma industry, fine, they should be able to use that. So we developed through sensing and through accelerometry technology, the ability to even deliver via a standard ISO vial. So we're very, very proud of this primary container agnosticity, and we want to impact the pharmaco's fill-finish process as little as possible. The second tenet of a really successful on-body delivery platform would be pre-filled and preloaded. Many of the solutions available in the marketplace today require that the device and the primary container be shipped to the end user, the healthcare provider, or the patient separately, requiring them to swab with alcohol the primary container, insert it themselves, and then initiate the infusion process. And this is tolerated, but we'd like to make this easier. And we'd like to be able to deliver a pre-filled, preloaded solution while being with as little impact to the pharmaco as possible, I should say. The third tenet would be flexibility in design and development. So we need to develop a platform so that clinical data, other data can be leveraged from that platform independent of which kind of primary container geometries and volumes you end up with. We believe that the platform should be easily customizable, where you can actually move between primary containers if needed during the development process. That was very important to us. And then the fourth major component is the user interface. It should be as easy to use as the simplest medical device that we can contemplate, which in our minds is a Band-Aid, right? So could we develop a solution where the IFU is remove the backing layer, place on the body, remove once it's done? So again, these are the guiding principles: primary container agnostic, pre-filled, preloaded, flexibility for our pharmaco device teams, and usability for the patient at the end of the day is perhaps most crucial. So I'd like to take a few moments to ask the first question. We're very interested in the demographics of the audience from the standpoint of, do you have a need for an on-body delivery system? So of course, those of you that are involved, that are from pharmacos, yes, we already have a product in development where we're super savvy on the topic of on-body delivery systems. B, yes, I've heard about it. We're thinking about it, but nothing to date, no immediate need. And the third would be no, absolutely not. So we'll give you some moments to cast your vote on these, and then we'll review the answers.
Thank you very much. And just a reminder to the audience, to participate, please select the answer that's relevant to you and then click submit. You can start voting now.
Game show.
We have 10 seconds left. Okay, let's see the results.
See the results. Okay, so a spread, really, a good spread as anticipated. Thank you very much for participating. That very, very useful. Okay, we'll continue. So let's look at these pillars of effective on-body delivery system design one by one. First being primary container agnosticity. So if you look at the image on the right, you'll see our platform device, 20 milliliter platform device, in this case, has a cartridge. The left-hand side of the device is where we pull the drug from. Okay, so this is the site where we have a needle carriage that penetrates the primary container and pulls the drug, which, by the way, we'll talk about a little bit later, but is very gentle on biologics, which is a very important consideration. The decoupling of the primary container on the left from the dosing cell and the componentry on the right means that we can change to whatever geometry and size primary container our partner needs. Again, we can develop in standard ISO vials, we can develop in off-the-shelf cartridges, we can develop in your primary cartridges. Why? Because of this decoupling, we can essentially make changes in the size of the outer shell via molding, very small changes to the fluid path, and you can go from a 20 milliliter to a 10 milliliter situation very, very, very easily. That was very important to us, right? That we have this platform that we can leverage clinical data from, regardless of what geometry and size primary container you end up going to market with. So a little bit more about this. Here you see a crude diagram of, the heart of the device, which is pulling from the primary container, in this case, a cartridge, and then the dosing cell on the right, delivering in steps of 4 microliters and allowing us to achieve within the industry standard of plus or minus 5% accuracy. Again, pulling the drug for lower shear forces on biologics. And because the principles of the fluid pathway remain complete the same across multiple sizes and geometries, again, we can leverage the platform and the data that we have for future submissions. Addressing the platform in a little bit more detail, you can see again on the left, you have this 20 milliliter platform device, and it can be configured to take a wide range of cartridges and vials simply by the molding of an insert, which keeps the needle axis on center and also closes the top of the primary container so there's no interference. There can be no interference by the user. So what you end up with is a platform that you could conceptualize as a 10 mil cartridge device, a 20 mil cartridge device, actually cartridges from 5 milliliters up to 20, all in the same device with no changes, okay? Only with the cartridge. With the vials, we go from 6R to 25R with this exact device without platform changes. So hopefully this gives you an insight into the flexibility. You could actually go to the clinic with that device and simultaneously be working with us on development of a different size outer shell, on the marketing, the branding on the outside of the device, and of course the things that we program in, such as the flow rate, the delivered volume, and the viscosity. So arriving at our second question, for those of you who sort of understand the current limitations of volumes that can be delivered, do you see an active need for 10 milliliter plus volumes delivered via either auto injectors or more likely on-body delivery systems? So again, same options of yes, no, or unsure. And again, we'll give you some time to complete this.
Okay, thank you very much. And again, just a reminder to the audience to participate. Please select the answer that's relevant to you and then click submit. You can start voting now. Okay, 10 seconds left. All right, I think we can check the results now.
Very good. Okay. Now that's reassuring, right? That we see almost 50% of our audience see a clear need above 10 milliliters. So we'll continue. Thank you very much. Okay, another example of flexibility. I spoke earlier about the need for proprietary cartridges in many cases with on-body delivery systems. Here is an example of some of the off-the-shelf products and suppliers that we have tested our device with. So again, we do not force you down a particular path in terms of primary container supplier, but we're very aware of the people that are leaders in this industry, and all of them work with our device. So again, optimal flexibility, we believe, for our partner. So the pre-filled preloaded challenge is especially interesting to talk about. And we'll start with the status quo, which is that two things come together, high level, for a combination product. So we have the primary container, which is typically filled by the pharmaco or a third party. And by virtue of their aseptic filling, we can conclude that the inside of that primary container is sterile. From a device standpoint, we usually see the device leaving the device manufacturer sterile also. No change in our case. Our device is a sterile barrier. So the device with no primary container inserted into it is a sterile barrier, with the only non-sterile areas of the device being the aperture for the primary container, the septum that the needle carriage sits behind, and of course, the outer septum of the primary container itself. So these things need to come together at some point in a way that maintains quality and sterility, in a way that doesn't impact cost significantly to the pharmaco, and in a way that disrupts the fill finish process and the final and the primary container insertion process as little as possible. So let's have a look at what the current kind of industry standards are for solving this problem. Well, number one would be patient filled at point of care, or indeed healthcare provider filled at point of care. So we see devices with essentially your reservoir is inside the device, and that device is shipped to the pharmacist, the primary care physician, or the patient with a needle and syringe, a vial, and a device with an internal reservoir. So again, in this situation, it's intensive on the patient or the healthcare provider. It is not so intensive on the pharmaco because they can use the same primary packaging that they've been using for decades, potentially. The second scenario, which we see, is that the patient loads the primary container at the point of care. So it's quite common. We see this with competitive on-body delivery systems. The device is shipped to the patient with the primary container, and the patient is responsible for ensuring the disinfection of the outer surface of the primary container septum. So we're asking the patient to take some responsibility for their healthcare. And in human factor studies that we've done, people are often not willing to take the responsibility, or they feel uneasy to take this responsibility. The third option is completely pre-filled, preloaded. So this is the holy grail from a user standpoint. They receive the combination product ready to go. So this is out there right now. But the one drawback, which we believe is significant, is that the pre-filled, preloaded devices require you to partner, in most cases, with a proprietary cartridge of either the device manufacturer or a third party. And we want to stay away from that. So really, we looked at these three scenarios and we said, what's missing? Okay, well, what if you could take essentially situation number two, but you could have a hypothetical worker inside the device that was swabbing those non-sterile surfaces with alcohol. So that's the fundamental question. How can you take that job of disinfecting those only non-sterile surfaces, and how can you insert that into the device? So this hypothetical worker inside the device, our team thought about this and landed on UV LED technology. Now, UV LED technology is very well established. And of course, this is super important when you're launching a product that you don't rely on super novel technologies, which people aren't familiar with. So UV has been destroying bacteria and other microorganisms for a long time, and it's trusted. Now, we are required to provide a log-6 reduction in certain types of bioorganisms, and we have repeatedly shown that this process does just that. So what's technically happening is you can see in the diagram on the left that there is an area that contains the non-sanitized components of either the primary container or the device. And that cell, which is completely enclosed with no danger of any of the UV leakage, that cell is irradiated. And after that point, our device knows whether that step was actually completed. And if it wasn't completed, the infusion would not continue. So this is what I mean by it's a validated process. We understand that if we apply a current of X for a time of Y, we are going to destroy and see a log6 reduction of all bacteria that the FDA are concerned with, including some UV-sensitive bacteria. So we've had several conversations with the FDA. The partners that we have in later stage clinicals in this platform, we've had to answer a lot of questions with the FDA, and they're very satisfied and moreover excited because they can move from a non-validated process of the patient swabbing a device to a validatable process of our device disinfecting those surfaces. And this all happens silently and invisibly in the first minute of the infusion process. So flexibility in design and development is also critical to maintain when you're working with pharmacos. And some of the things to consider, what's in our toolkit to give our partners flexibility? Well, first would be the software-enabled electromechanical pumping mechanism that we spoke about. This enables us to program any infusion profile that you can think of virtually. Okay, so this allows us to have ultimate customization on the flow rate, the forces needed to drive the viscosity of the drug, and the infusion profile itself. So from a development standpoint, these can all be changed during the development cycle, and it's these programmable inputs that we're optimizing as we go down the drug development pathway with our pharmaco partner. Smart sensing is very, very essential in a device of this sophistication. So we have occlusion detection, we have air detection, we have an accelerometer that keeps people in orientation compliance for the vial-based device, for example. And then lastly, we have connectivity, both in the form of NFC, which is the same very, very secure technology that your Apple Wallet, for example, uses. And we're actually burning the software on. It's custom for every single drug product and pharmaco that we interact with. And then on the other side of the infusion, you have Bluetooth that allows the event log of everything that the device did. And that can be sent to the pharmaco during development, or a much higher level version of those details could be sent to a healthcare provider immediately after infusion. So they could receive information like, did the patient rip the device off their body halfway through infusion and cause the device to error and for the needle to retract? Well, we'd know exactly how much drug was delivered, and we'd know exactly what the problem was with the compliance of the user at that case. So this is our toolkit, and these are the things that we use when we think about flexibility of design with our partner. So in a little bit more detail, the electromechanical pumping mechanism allows us to basically step the drug into the patient 4 microliters at a time, allows us to achieve plus or minus 5% accuracy, and interestingly, up to 3 milliliters per minute of infusion. And of course, there is a trade-off between speed and the viscosity of the drug, and we'll talk about that on the next slide. But up to 3 milliliters per minute is validated. Various flow profiles, we've spoken a little bit about that. You can see a stepwise infusion in the bottom example versus a steady state. Other examples would be delayed start, okay? Would be bolus initiated by a user pressing a button, a standard basal and a bolus initiated by the user. We could have intermittent delivery, and again, we can have steady state obviously as well. So we like to say, if whatever infusion profile you can dream up, we can program into our device. Okay, so as promised, you can see here a little bit of the relationship between the flow rate and the viscosity. Up to around 20 centipoise, we can deliver north of 3 milliliters per minute. As the viscosity of the drug increases by around 180 centipoise, we have to limit to 0.5 milliliters per minute for safe, effective infusion to occur. Now, 180 centipoise is not a limit of our technology. It's the limit of what we've been asked to review. So we're not sure. It could be much higher than this. Please challenge us. That would be another message. Please challenge us with this, because we are the company to address your needs regardless of viscosity. A little bit more detail on the fluid path and what allows us to work with both a vial and a cartridge. You can see to the left there, the top picture represents the slight change that's made when we have a vial-based primary container. So of course, as we pull out drug, we need to equilibrate that closed space with clean air, which is drawn from inside of the device. The difference with the cartridge, you don't need that. With a collapsible cartridge, you only need one needle. The rest of the fluid path remains completely untouched. So let's focus on usability. Again, I think it all kind of culminates to simplicity of use at the patient. And we'll be playing a video here that I will endeavor to speak over. We'll give you an idea of what the patient experiences. So the first thing that the patient does is, well, they have access to a viewing window. Very, very important that the patient be able to see the drug is effectively being infused. First thing that they do is remove a backing layer, which wakes up the device, and then the device is placed against the skin. The user initiates the treatment, and that activates an on-body needle, 5 millisecond deployment. It's faster than your brain can register the pain, so we call it virtually painless. The infusion progresses after the UV disinfection cycle. The infusion commences. We go through 4 quadrants of delivery. Then the needle is retracted, and the data of the infusion is sent to whichever parties the pharmaco deem fit. And of course, it's very, very important to us that we help people with chronic conditions live a life as close to normalcy as possible. That really is our North Star at the end of the day. And a little information about the array of primary containers. Okay, a little bit more about usability and the fact that we really do view this device as close to a Band-Aid as you could get, especially for such a sophisticated technology. It really is as simple as peel, stick, remove, and discard. So we're very, very proud of the fact that we've engineered a solution around this that hopefully helps both the pharmacos and the patients. A little bit about the user interface. So we have an adhesive backing, which has been innovated to allow the patient to have movement, but at the same time for the functional part of the patch to be completely adhered to the patient, resulting in 0 wet infusions and almost no irritation at the patient level post-infusion. We have a very simple interface, and this is by design. We have a button to press to start the infusion, and we have tactile, auditory, and visual sensing, display in order to allow the user to see how far through the infusion are they. So, for example, if we got 3/4 of the way through the infusion and the patient caused an error in the device, such as removing the device prematurely, you would get a high-level take on how much drug has been delivered, and then the event log of the device would capture exactly how much had been delivered, and then we can subsequently relay that information to a healthcare provider, or if it's during development, we can allow the pharmaco to see that data. Again, a note on connectivity. We have both Bluetooth and NFC. NFC is essential for getting the software onto the device, the software that's customized per application, and Bluetooth is there if needed. We typically don't get asked by pharmacos to develop apps for them because they have very strong teams to do that. We will interface with a pharmaco to work on apps if needed, but we haven't found that to be the case so far. So the technology is there, but we do not have an opinion on what you do with that Bluetooth technology. That is down to the pharmaco and the information level that they want to share with whomever. and of course, a high degree of cybersecurity involved with this device, and much data to share with future partners. So none of this is theoretical. We also have scaled up some world-class manufacturing facilities over the last several years. We've inaugurated several ISO 7 clean room contained manufacturing processes, 3 lines since 2019 and more to come for the rest of this year and beyond. That allows us to achieve a capacity of around 1.5 million units. And that's the horizon that we see in terms of clinical numbers, right? So for our folks that are in the clinic with us right now, we have more than enough capacity between the 1.5 million units we can make today and the additional 1.5 million units that could be achieved via further automation. Now, on top of this, we have a very dialed in, very efficient manufacturing process that can be scaled either on site or in a different location in Europe, North America. These are all thoughts on our mind right now. And it's great that we've dialed in such an efficient manufacturing cell that can be scaled at need. Okay, next question. Will we continue to see growth in subcutaneous administration versus IV? Not a volume question. This is really more about pipeline drugs and whether or not we're going to see things developed, as I see, more subcutaneously than via IV. And again, yes, we do. We think that there's going to be immense growth in subcutaneous with all the reformulations. No, we don't think so. We think that there is still a huge need for IV-infused drugs, or C, I'm unsure. I don't know. And we shall give you some time to respond.
Yeah, thank you very much, Greg. And just a reminder again, to participate, please select the answer that's relevant to you and then click Submit. You can start voting now.
We should have music playing in the background, probably, yeah.
Okay, I think we can check the results now.
Okay. Nice. Okay. We're in violent agreement. There is a need for continuous growth in subcutaneous administration. This is very nice for us to see, and thank you very much for participating. So spoke a little bit about growth capacity. Again, we can get to 3 million units by further automation of our current processes, and we have a very scalable manufacturing cell as volumes are required in excess of 3 million units. So what do we do in production? We've developed a combination product with you, and we are going into production. Well, we handle everything, of course, from raw material receiving, through production, through packaging, through shipping. Through our acquisition by LTS, we have access to sites that will be able to insert primary containers into the device. The only thing presently that we cannot do as part of the end-to-end solution is the filling of the primary containers, which is usually done by the pharmaco or a third party. But we also have an extensive network of folks that we can align you with so that you have a full end-to-end solution from the device production to the filling to the insertion of the primary container in the device. Okay, so lastly, I'd like to talk about what it feels like to work with us, what you can experience, what you can expect working with us. Well, we're not a device manufacturer. We are, but that's not just what we are, okay? We believe that while it's your submission and your drug as a pharmaco, we're responsible for supporting you in that submission process. So here's an example from development to post-market of some of the things that we commit to as part of our development programs that we sign with partners, all the way from design inputs through IND submission, through BLA submission, to post-market surveillance. So we see our responsibility as much more than providing an easy-to-use, easy to change and configure device. We are with you until launch and beyond. And then I think, lastly, I believe, this is the structure of how we interact with our customers. we have a milestone phase gate process, which is probably familiar to many people. And we certainly are no exception. We start at phase 1 with a feasibility study, which I like to think of as it's just a collection of experiments that the pharmaco determines are needed to de-risk the platform in their eyes and really make it easier for them to move into the first stages of real development. Conversely, on the other end of the timeline, we have design transfer. So we are actively involved in the commercialization and scale up of the product. Now, even within this framework, there is flexibility. We have had partners in the past who have said, we think about it a little bit differently. So perhaps they have their user requirement phase in their feasibility phase combined into one study. We're very flexible about the experimentation needed to de-risk our platform in the mind of the pharmaco. So even within our structured process, there's a lot of room for maneuver depending on our pharmaco's needs. And that is it for my presentation, and I would be absolutely delighted to take questions, and I'll be reviewing them in the question box. Thank you very much for your attention, and I hope we managed to answer the question and give the fundamental takeaway that we have engineered our way around the compromises that pharmacos and patients are currently being asked to make. Thank you very much.
And thank you very much, Greg, for this presentation. Just a reminder for the audience to ask questions. You can send them via the questions widget. Just type them in this box and click submit. Please note that you can still send your questions after the webinar and they will be answered at a later date. And it looks like we've got several questions already. Okay, and our first question is, what is the size of the 20 milliliter device?
So we can send specifics, but hopefully you can see this. This is an iPhone 13 Max, and you can see the size of the device relative to it in the profile. So as with all the questions, we can send specifics to the audience afterwards for any questions that we cannot get to, but I can at least show you a visible, representation of it.
Okay, thank you very much. And our next question is environmental considerations of the device. How do you dispose the device, and can any parts be recycled?
The answer is yes. We work with our pharmaco partners. Of course, getting the device back as a fully disposable device is a challenge that many device manufacturers and drug companies are having. But once the device is back in our possession, we have third-party providers that we've partnered with that can open up the device and can optically sort the battery from the PCB from the recyclable plastics. So ultimately, we have a solution that can lead to recyclability of the device, and we have to partner with our pharmaco to find ways to get that device back. And I think that is an industry challenge right now, to be completely honest with everybody.
Okay, thank you. And our next question is, must the devices be shipped to us for filing?
For filling, no. In terms of insertion of the primary container, we can receive the drug in its primary container, and we can load it at our site. So it's really what is best for the pharmaco partner. What we cannot do is fill the primary containers, but we have a third-party network that can help with that.
Okay, thank you very much. And our next question is, how can you know the UV sanitation step occurred?
The device is kind of self-monitoring. So essentially, we know that a certain current applied to the UV cell for a certain amount of time, 30 seconds, is sufficient to kill the bacteria that are of concern to ourselves and the FDA. So we actually have an overkill cycle of 1 minute, and the device checks itself to make sure that that step of the process occurred. It makes sure that there was a current draw and that was applied to the UV cell for a certain amount of time. And if that does not occur, then the infusion cannot proceed. Hopefully that answers the question.
Yeah, thank you very much. Okay, and our next question is, you stated the device is gentle on biologics. Can you elaborate?
Yes, I'm certainly not a physics or a fluid dynamics type person, but I do know that pulling on a biologic is much gentler, much lower shear forces than pushing, and that's the reason that we chose that mechanism. We've tested over 80 biologics of multiple molecular sizes and of multiple sensitivity, and we have never failed on bioavailability of the drug after it's released from our pump. So that I can elaborate a little bit and share that with you. For more specific data, I can reach out to the questioner offline and give them a more comprehensive answer.
Okay, thank you very much. And just a reminder that you can still send any questions while watching on demand. All questions will be answered at a later date. I would like to thank Greg for this presentation and LTS for sponsoring this session. To the attendees, you will receive an email telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is bmi-live.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. Thank you once again, and I hope you all have a nice day.